Thank you. Um, Biological databases and integrated information retrieval. How we can get the information and the word I use integrated, you will understand when I try to show you the integration of the different databases in the mainly NCBI. Okay, so outline of this today's lecture. So we'll try to once again understand some of the basics, information about NCBI and different information retrieval system from NCBI, GenBank flat file features and FASTA features, and how to retrieve nucleotide sequences from GenBank and ENA, protein sequences from GenPept, that is NCBI one. When you have a nucleic acid sequence, we call as GenBank, and the nucleotide amino acid sequence we call as GenPept. And other databases of NCBI, like as I told you, integral system. So previously they used to call as NRES, and they are not calling that as NRES. So still the function is the same. So I will show you. Then PubMed, OM, IM, taxonomy, genomes, and few more are there. We'll see. And they did PDB, how to get the data. KEGG, once again, we see some of the information. And UCSC Genome Browser, this is another interesting, browser for many different purposes while analyzing the genes and their features. So I want to show you today. And 1000 genome browser. It's not 1000 genome browser. So it's not the $1,000 project. This is different. Don't confuse with that. 1000 genome browser. So I'll show you what is that. Okay. So NCBI. This is present as you can see Rockwell, Epi, and Bethesda in USA. So after recognizing the importance of computerized information processing methods for the conducting biomedical research, the NCBI was established on November 4th, 1988 as a division of NLM, that is National Library of Medicine at National Institute of Health. NCBI creates public databases, conducts research in computational biology, develops software tools for analyzing the genome data, and disseminates the biomedical information, all for the better understanding of molecular processes affecting human health and disease. This is how it looks like, the home page of NCBI. Okay, so like yesterday, in between the slides, we will go to the browser and show you. So instead of waiting until the finish of the slides, so wherever it required, I will show the browser to you. Databases, NCBI. That is, NCBI assumed responsibility for the GenBank DNA sequence database in 1992. And in addition to GenBank, NCBI supports and it distributes a variety of databases for the medical and scientific communities. These include Online Mendelian Inheritance in Man, OMIM, Molecular Modeling Database, MMDB, 3D Protein Structure, Unique Human Genome Sequence Collection, UniGene, a gene map of a human genome, a taxonomy browser, the Cancer Genome Anatomy Project. These are all few, there are so many. Maybe you can see when we are browsing. And come to the next topic, nucleotide sequence databases. And mainly these are the three: EMBL, GenBank, and DDBJ are the primary nucleotide databases, a major one, and publicly available, open access. It is called as freely available. These are the links to go to those. Yeah, to obtain DNA sequences, you can check these three. And as I was discussing with you, so this NCBI EMBL, that is EBI, and DDBJ's CIB. Those three, the GenBank, EMBL, and DDBJ, their databases, nucleotide databases, or sequence databases, either nucleotides or amino acid sequence, will be interchanged within a day, within a 24 hours. Uh, so wherever you submit in the NCBI or DDBJ or EMBL. So within 24 hours, uh, they will update the information by exchanging the information as part of the database collaboration. So how to search the nucleotide sequence in GenBank, that is NCBI. So you need to first select as a nucleotide. There are so many uh, tools available, tools or databases available in this menu. And then you have to type the name of it, what you are looking for. So there are two ways you can search. You can use a crude name of it and put search, then you will get a multiple results. So you see you can get here 1000 to 11,117 because the whatever the name based on this name there are so many results are present. So it may not be accurate, but you have to screen which you want. So maybe you can go with a virus, then you can search there. So or you can improve your search by adding some more information there using the Boolean characters that we will see later when we talk about the PubMed. The best accurate result is give the accession number as we were discussing before. Accession number is a unique to each nucleotide entry in the GenBank database. So once you give this one, the one it will give you the automatically it will open it, not even give the possible outcome. So it will open the output of the particular the accession number. So this accession number is the nucleotide entry, the name of it, severe acute respiratory syndrome, coronavirus, 2 isolate Wuhan, 1 complete genome. This is the name of it. And this is the format how it will display in the GenBank. So this whole format can be divided into 3 parts. This first few information is under header, and the middle one information is the features, and the following by the sequence. Though this is also called as GenBank flat file format. This is also called as flat file format. So this is also called as GenBank flat file format. So if you go individually, the header, this header contains, these are the informations as discussed roughly earlier. This is having accession number, which is unique to the each sequence, and version also will be there if there is any update. So accession number won't change, just it will be added extra digit decimal after the dot. So see the version is 2, so NC it is masking, so NC underscores 045512.2 if it is any different versions. And this is the access number, this is the length of it. So how much is the length? This is the 3822 base pairs. This is the length, and molecular type is single stranded RNA, as the genomic information for this virus is a single stranded RNA and is a linear. And the molecular division also will be mentioned in this flat file. That is VRL is a molecular division stands for the viral, and the followed by this information is a modified date when it was modified. It's July 18, 2020. And apart from that, some more informations present here, the source and taxonomy of it. So this is the coronavirus 2 is a taxonomy. You can see under the virus, Riboviridae Arthurnavire, Pisuviri, Kota, and so on. And finally, the family is Serbico virus. Under that, this is a coronavirus. Okay, so this is the first author who published this information in 2020 in Nature, and followed by supporting publications will be present here. So that is about the header information of this flat file. And these are the divisions. So as mentioned here, the molecular divisions, what are the divisions we do have in general GenBank divisions? So BCT stands for bacterial and archaeal, and INV is invertebrate, MAM is a mammalian origin. So sometimes, if they are very particularly present as rodents or primates, it will be separate rodent and primate. But other than that, it will be considered as mammalian. And Faj origin is PHG and so on. So viral, as mentioned just now in the flat file VRL, is a viral. So other vertebrates, it will be present as a VRT. That is the traditional ones, and some more we do have bulk one. So like a patented sequence. Okay, so it is not mentioned what is the origin, just patented. So P8 it will be, and sometimes expression sequence tags, as discussed earlier. Some of the cDNA sequences, sequenced cDNA sequences. So they are mentioned as expression sequence tags, so that whole library will have few thousands of sequences. So they generally put as EST. Sequenced tagged sites or genome survey sequences. So these are all short fragments of the genome sequences, and not related to any protein coding gene or any gene, just some survey sequences. High throughput genome. So that is now more popular. So whole genome sequence as a high throughput. So they're submitting as a high throughput genome sequence. So they put as a HTG if it is a genome, and the cDNA is a HTC. And context. So we have seen what is mean by contig in the first practical. So you sequence a few, I mean one gene or one DNA segment, but you sequence multiple sequences, and you align it, and you get one contig. So that contig is you generated from the few sequences which you sequence from some origin. So those you can submit as a context. If you get multiple contexts, you can submit as a multiple context. So that is about the header, and the middle one is the features, and this contains, first one is of source, and it is 1 to 3822. So this as a simple small file, it's showing as a one whole sequence belongs to the only one gene, that is gene S. So I'll show you later when we go through the browser different examples where you will have multiple genes and multiple CDS. So this whole 3822 are coding for a protein, spike glycoprotein. Okay, so this is having its own gene ID. This is also unique ID. Okay, this is a reference number is 343740568. So here we are searching in the nucleotide database. So this ID you can give in gene database. So genes also have its own ID. Okay, you can get the same the spike glycoprotein information. And CDS coding region is from 1 to 3822. There is no exons or introns because this is from the viral origin. And this protein, S protein, and the sequence, once it gets translated, what is the translated amino acid sequence also will be displayed here. Just a minute.